Am I audible?
Yes, you are audible, ma'am.
Very good evening to everyone over here. Today I have been given the topic Analytical and Forensic Toxicology. It's a very interesting topic in the sense that if you want to study about forensic toxicology, we should be knowing about analytical toxicology. Uh, in nutshell, if you want to say, if you know the analysis of components present over small components, let it be metals or any genoetics, if it is present in the blood or urine or your gastrointestinal contents over here, then you are able to know what is the condition of death of the animal or suspicion of any criminal offenses done to the animal or our human beings also. Actually, generally we are veterinary toxicologists, so we will talking about the animals more. But generally, here I can talk about both the things: medical terminology and veterinary terminology. Okay, we'll go to the next slide.
Yes.
Okay, as we were discussing, so analytical toxicology, first we will understand about analytical toxicology. Here, as we know, when we were doing some research work, there are many applications of analytical tools. So generally, we have the equipments where we are able to quantify or qualify the amount of materials that we are suspecting, what is present or what is not present. Generally, in case of our toxicology cases or the cases in which there is some suspicion of some toxicity, there will be some amount of genobiotics. Okay, so we need to learn or we need to assess the presence of those genobiotics in the whatever sample is being presented to you. For that, we need to have some analytical tools. Tools. In these analytical tools, we should be able to estimate it qualitatively or quantitatively. The term qualitative means either it will be present or it will be absent. If it is present, means if you are searching for, see, if you are suspecting for, let us suppose talk about arsenic poisoning. It is very prevalent in this coastal belt. So if there is arsenic poisoning, we are searching for this amount of arsenic being present. So if we are going for qualitative set, we will see if the arsenic is either present or absent. It should be present or absent. Or it is about I estimation, in which you can give double plus or single plus or triple plus. It is present in that amount. But when we talk about quantitative estimation, then there should be a figure. There should be a figure of 0.29, 0.0024, like that. So that is quantitative estimation. So we should understand what is qualitative estimation and what is quantitative estimation, and their estimation when it is present in the body of the animal or the sample being presented to you. So generally, we are searching for some genobiotics or some outside material which is present. Okay. So this genomatic, what it does, this genomatic, suppose it can be a metal, it can be a non-metal. Sometimes it can be some plant toxin products too. It can be also some your soil pollutants or air pollutants. So anything which is not normally present in the body, or anything which is not a part of our body, or not a part of your nutrition or the food which we are consuming regularly. So it is a genobiotic, and it is going to have some deleterious effect on the organism or on the human being through its metabolism action. Whenever any genobiotics is going into your body, it is going crossing through your liver, it is undergoing metabolism, and then it is showing its effect. After this metabolism, whatever products are coming or the byproducts are coming of the genobiotics, they are going to produce some harmful effects. So it becomes a toxin, and we are able to know that some toxicosis or some toxin is present, some toxicosis is being present, and some signs and symptoms are being shown by the um, organism or by the human. So what is happening when we are going for this analysis portion? So we need some samples, or if the animal is the human being or animal is that is presented to you is alive. So in that cases, we can take the blood samples, or if it is a postmortem or death has occurred, we can take the postmortem samples. In that case, we can take the tissues from the liver, or generally gastrointestinal contents they can be taken, and in that also we can take the find the by the analysis we can find the genobiotics. So both we should be able to know if the present form of genobiotics is present or it has been metabolized. Once it is metabolized or some hours has passed, it will convert it into some metabolite. So that metabolite also we should be able to recognize. First of all, we should recognize the genobiotic itself, or if at the time with the passage of time it get converted into different metabolites. That metabolites also we should be able to know. Okay, by knowing those things. Next slide, please, ma'am. Ma'am, next slide. Uh, By knowing those components, what is present, how much is present, we can able to know if at all some deleterious poisoning or malicious poisoning or accidental poisoning has occurred. If the poisoning has occurred and we are taking it for the purpose of law, we are taking it to the law court that of course this poisoning has occurred and this much has poisoning has occurred, but this material has been consumed by the human being and that has occurred, and we are taking it the purpose of law for punishing some person. So for that, we need very stud information that okay, this thing has occurred. So when we are going to the court of law, it becomes forensic toxicology. This forensic toxicology and analytical toxicology, they go hand in hand. Once we are able to analyze the presence of toxin in that, then only we can give the permission or give our witness or that okay, this are present in there. So it might be that this kind of poisoning or this toxicity has occurred. Okay. Many times we also see go for regulatory toxicology. It occurs in the workplace. Many times in the workplace also we are able to detect some drug or some other genetics or some intentional poisoning. It can be also causing some deleterious effect, like injury will be there to some organs of the body. It may be internal organs or external organs, or some deaths may be there, or any damage should be there. In such cases, we should be able to know how the toxicity has occurred, why it has occurred, what is the concentration of toxin that is present over there. So many times, if you are able to know the evidence that okay, this is present positively with this much amount, so we can prove it, and we can also prove it in the court of law. So that becomes forensic toxicology. So we should be able to understand what chemicals or genobiotics can be present over there. Suppose we are going for an industrial area. So there we can suspect the presence of some metals or some non-metals from the metallurgy unit. If we are going to some workplace where they are going for this asbestos refining or something like that, so we can suspect that thing. Many times it happens that in the water also we are getting the toxins. If we are going for that area, if the soil or the environment is having those amount of those toxins, so we can carry out tests for those toxins over there present in that. We can suspect okay, this area is prone to this type of toxicity, and we can suspect, and in the sample we should try for those toxins, and if we are getting positive for those samples, we can say this is present. Okay, ma'am, next slide. Generally, if we are taking the samples, if the person is alive, we are taking the urine samples. Okay, when we are taking the urine samples, we can test it for the presence of any drugs or any suspicion that we are having, and we can sort it out and give the result. So by knowing this diagnosing that okay, these symptoms are being shown by the patient, and these are the present. When we are going for diagnosis, it is generally first of all it will be symptomatic diagnosis, and we go for symptomatic treatment. We are saying these symptoms are present, so we give symptomatically, and then we go for the analysis. After analysis, when we are getting a specific toxin present over there, we can go for the specific antidote and we can treat the patient. So generally, when we are going for this and our analytical toxicology, we are able to detect those drugs. So generally, we need that those amount of drugs or the genobiotics, they should be rapidly moved out from the systems, for which we allow some emetics to be given, some purgatives to be given. Along with that, we give our fluid therapy so that to dilute the toxin or the poison over in the body. Generally, occupational toxicology and regulatory toxicology are two terms. Occupational toxicology is the toxicology which occurs where the person is working. If it at all he is working in a suppose this asbestos mining portion area or mining area, the way he works, he will be having some infiltration into his lungs, or if it is going through orally, it will go to the liver. So generally with the liver and the lungs or the urine, because kidney is also a organ through which it is being metabolized. So these are the organ through which the toxin goes or the xenobiotic goes. So we will be generally taking the blood samples or the urine samples, or if in case of postmortem, then we can take liver pieces also to find out the presence of those toxins or xenobiotics over here. Next slide. Next slide, ma'am. Okay. When we are going for dosing of the monitoring, so if we are treating a patient, okay, we have got the patient and the patient was suffering from some toxicosis, and we have given them whatever I told, like this MSC, so we have done, and if I given some laxatives, and we are also giving some atropine sulfate or something like that, we can monitor the amount of toxins that is present. Slowly, by the process of detoxification, the toxins will get reduced, and by constantly monitoring, we can get this toxin is being reduced from time to time. Also, the viability we can check from time to time, and it can be a important parameter. Viability is the amount of toxin or the amount of external genovitis which is present over the over period of time. For this, we should also understand how the pharmacokinetics has occurred, or how the drug has been, or the drug or the toxin has been absorbed into the body. That is the thing that as we talk about the pharmacokinetics of any toxin also, we go for ADME, where there is absorption. If at all it is a inhalant type, or it is a toxin in the environment, it is present over there, so it can go through your nasal system, it can go to the lungs, and it can happen to occur in the lungs. Or else, if it is being topical applied through screen also, there can be absorptions, and oral absorption is always there, and it is metabolized by the kidney, and it goes to the blood circulation. Distribution is the process where the drug has gone over there, a drug or toxin, or any metals or non-metals, or any toxin, it has gone over there. It is going through the blood vessel and getting distributed to whole the body. Then it goes to the process of metabolism or the liver, where the liver undergoes the process of biotransformations. There is phase one biotransformation, phase two biotransformation, where we are going to produce one soluble metabolite, and it is easily excreted away from our body. So from every point of time, if you are able to measure this amount of toxin, and we can able to know how much the toxicity has occurred, or how much the patient has recovered. This we can be measuring over the period where we are able trying to recover the patient from it. So forensic toxicologists, they are using this analytical chemistry to understand these hazards of the toxic substance, as well as the toxic effects which is coming out of it, as well as how to recover from it. If at all the patient is recovering, or how much time it is taking to recover from the condition. Next slide. Okay. We will talk of a condition where there when this condition came, that how we will know how the toxicity is occurring, how the analysis can. So first, what is the thing when there is toxicity? So we should be able to separate the toxic agent. When we want to separate the toxic agent, first, when the starting period, they were not at least able to know that if the patient is dead, or we can able to think the toxicity has occurred out of another thing also. If you are not able to see the symptoms, if you are not able to get some estimations, apart from that, if the metabolite is there, if the urine sample is there, from that also we can get. So slowly over the period of time, this came into being, that if at all we are not able to see the patient itself, or get the sample from the patient itself, we can see other things also. Like for example, if the parent compound is not present over here, we are not able to get the parent compound in the blood sample, we can go for the metabolites. So we should be able to understand which compound he has given the which metabolite. By seeing the metabolite also, we should be able to understand which toxin was there. Also, many cases, the gases will be liberated. Suppose in case of carbon monoxide poisoning, the patient must have inhaled that carbon monoxide. So if at all we are getting the lungs, now we can squeeze out the lungs and we can get the gas, and we can go for its specific test, and we can know that carbon monoxide poisoning has occurred. So this carbon monoxide also, it will be acting throughout our blood circulatory system, and there will be formation of carboxyhemoglobin, and there will be color change of the our hemoglobin, and we can understand from that process also. Next slide. Okay, there will be many kinds of toxins also that we're talking. I was telling that can be metals, non-metals. Also, that can be corrosives. As we say, the corrosive means which it can cause some corrosion type of thing. There will be deleterious effect. And also we guess the metals and the non-metals, like our metals will be arsenic, cadmium, your all the heavy metals, and non-metals like phosphorus, fluorine, chlorine, even salt also causes toxicity. So these, when at all they are going into the body, they will cause the symptoms. The symptoms comes out of the change in the blood biochemistry. If at all the blood has been changed, so there will be changes will be there. So metals generally they are going into the body of our patients through occupation or the environment. If the environment is having the if the environment is having those amount of metals in it, so slowly it will get into the body of the patient through the day-to-day life. If at all the place of occupation is having the concentration of these metals, that can go into the body of the animal or our patient. Another thing is that if there is environmental hazard, many a times you can see that there will be some smelting plants or some steel plants or some plants will be there, and they will be releasing those extra their corrosive metals into the river itself or into the atmosphere itself. So many times we see that the atmosphere gets wholly polluted, and the people living over there, they are taking these toxins over low concentration for long period of time. So slowly it will go into the body and they will localize in the body of the patient, and many a times it will give rise to the different metabolites, and it will be harming the body of the body. So many times we can see one compound like, for example, here it is stated mercury. So mercury will be having metallic mercury, mercurous iron. So all of them are not having equal potency of toxicity. It will be different, but it the more toxic ones, it will be showing more toxic symptoms, and which are not toxin, lesser toxic, will be showing lesser symptoms. Moreover, if many a times we can see it is in anionic form. So ionic form, they can be more toxic. So even if phosphorus is there, it is having two portions, white phosphorus and phosphorus, and one is more toxic, one is less toxic, like that. Many times organic substances also, they are also problematic, and it can be your natural products. Many times we have seen some plant toxins will be there. Plants will be toxic, like your Nerium oleander is there, your Cannabis sativa is there, your Calotropis is there. That cannot be deliberate poisoning, but many times toxins are given deliberately, or some suicidal tendency, people like that, they consume that. Apart from that, pesticides are there for suicidal purpose or homicidal purpose, people may take that. Also, there are many accidental poisoning. Pesticides they have kept somewhere, and it is getting into the hands of the people when they are working around it, and they are just ingesting, or they are making the food. Along with that, it can go into the body. In such cases, we can also get the extraction of such in the body fluids also, this concentration comes. So if we are getting this, we are getting this body fluids, we are taking the blood, and we can analyze it, and every time we can get those corrosives or metals, non-metals, and we can have it. Next slide, ma'am. Excuse me, ma'am. I will put this 1 minute, ma'am. Am I audible, ma'am?
Yes, you're audible, ma'am.
Okay, thank you. So many a times we can see and identify the purity of the compound. See, we are able to know this toxin might have occurred because this is the place where the pollution was there, or the smelting plant was there, or many times we are getting such kind of toxicity over there. So we can see that this can be there. For that front also, we should be able to know how much toxin has got into the body of the animal. For that, we need to identify or purify the compound, how much concentration of that genobiotic extra has got into the body of the person. For that, we need to do it the study very accurately. Many times we are also seeing that drugs also, or the doses or the sedatives or something is present in the households, and people are taking it, and that is also showing toxicity. Also, we can see this 2,4-D, or these are herbicides, and these were being taken used as toxins. And many times the presence of one compound makes another compound more toxin, and in such cases, bigger amount or more toxicity occurs. But one of the very important things of analytical toxicology, we can be able to monitor the doses or the stability. See, if many cases it can occur that the toxin was present over there for a particular period of time. After that, the toxin has been removed or the person has moved to another place. So in that case, the toxicity will decrease because the person is no longer getting exposed to that toxin regularly. But in case of some malicious poisoning, but in case of some wars or something like that, so huge amount of toxin must have entered to the body of the animal, and this have gone into the body of the animal and interacted with its body parts or the body fluids, and showing many different toxins or toxicity symptoms over there. So if analytical processes over the period of time we can develop, through which we are able to recognize what the compounds are present, what components are present, and we are able to know what toxicity occurred. So we can be able to know the result. Okay, if this toxicity has occurred and this treatment we can start all of a sudden. Next slide. So, in the generally in general toxicology, what will be the role of our analytical and forensic toxicology? Analytical methods, it is needed to understand the bioavailability of the compound. Bioavailability means how much portion of that compound is present now in the body. Okay, so if the compound is having low water solubility, so it will be lesser spread into the body of the animal. However, if the blood concentration is it is having a vehicle, so it will be less dispersed. So if it is having a oily vehicle, so it will be effectively absorbed throughout the system, our body systems. Next slide. Also, there is a dose response relationship. I also describe to my students what happens whenever the dose increases, the response increases. In the same way, if the dose is going on increasing, the toxicity is also going on increasing. But it will go on increasing to a certain level until it has occupied all the receptors or occupied all the present over there. After that, it will show the highest toxicity. After that, it will go to a plateau. And if the toxin or the source of the toxin is removed, then that level of toxin can be decreased over the period of time. If you are giving the therapy over a period of time, that can also decrease slowly over the period of time. After in case of our postmortem examinations, we are doing this qualitative and quantitative analysis. After the collection of the samples from the postmortem hall, we can see what are the effects it has shown. So generally, when we are going for the test, first of all, as we are told, this qualitative test will be there, quantitative test will be there. Also, if we are taking the blood sample, we can see also the harmful effects it has done to the different body systems, like how it has affected your blood system, like the blood profile is there. We can go see the amount of hemoglobin. If it has decreased, we know the normal value. How it has deleteriously, it has decreased your hemoglobin level or PCB or MCB. Those levels will be. Also, we test for the serum biochemistry. That is the way we go for the liver profile test or the kidney profile test. As liver and the kidney both are organs of where the drugs are being metabolized and going, and also the kidney where it is being excreted. So those two organs, if we are seeing, we can see the deleterious effect more. And the liver enzymes like the HDLT and the kidney enzymes, that the urea, creatinine, bull, they are the main indicators of toxicity. By seeing those parameters, we can be able to know how much toxicity has occurred and what level the toxicity has occurred and how much it has occurred. Also, in case of postmortems, we can go for histopathology or the conditions in which we can see, take the tissue samples and process it and see what deleterious effect it has given done to the tissue level. At the tissue level, if there is deposition of some sort, yeah, in many cases we can see the deposition. Calcification is seen in case of heart tissue, and also in the kidney tissue, that the nephrons are how they are destroyed, or any deposit has been in around the nephrons or that is the way we can know that okay, this type of toxicity has occurred. So by seeing all this data, or then if we have got the sample and the sample is having a good concentration, we can take and we can isolate also. We can isolate, purify, and we can go up to the level that we are able to know, okay, this specific toxin was present, and we can designate that this caused the death of the animal. Next slide. Also, there are circumstantial evidence. Circumstantial evidence means whenever the death is occurring, what was around in that area. If at all the patient has died of something, suppose we can say talk about some pesticide. If the patient has ingested the pesticide, it might be happened that in his or her circumstances some fight or some problem has occurred, some suicidal tendency is there. So if we take the history of the patient, that how what was the previous occurrence in that case. So we can say that according to these circumstances, he might have developed a suicidal tendency, and we people know that this pesticide consumption can lead to death. He or she might have taken that. Many times we are seeing this ethanol is being consumed by people. Many times when it is going to higher level, or many times we can also see cases where there is mixing of other compounds apart from the compound that the person is taking. Other compounds might be mixing, and those compounds also take toxicity. In those cases, it is not that the person was suicidal. He or she was only thinking that I will get some intoxication for some time and I will get out of it. But it is if there is some mixing of some other compounds which is more toxin, and in those cases also we can see deaths are there. Many times we can also see some designer drugs are there. Those drugs they are giving rise to more toxic or more deleterious compounds. Those compounds can cause more toxins, because nowadays we can see many types of analogs are coming which are designer drug, or this ecstasy type of drugs, this GSB type of drug. This has been consumed. When the person is taking that, he or she or taking that, I will be intoxicated for some time and then I will get out of it. But the problem occurs when it is getting mixed with some other compound, which is more problematic or deleterious or even death-causing type of compounds. Those compounds, when they are taking, there is accidental death in many cases when the person was not intending and he was he or was not able to know what will happen. Many times we are also saying that we are taking this analgesics when we are suffering from this, we are getting some pain, we are taking opiates or analgesic. But the thing is that a certain time, after certain time, these painkillers also we get habituated. And when we are getting habituated and we are taking it for long period of time, it also becomes deleterious. And if the concentration of drug is remaining in your body for a longer period of time, it can also be harmful. Next slide. Okay. In being forensic toxicologists, we need to go and tell the court of law that this is the cause of the death. For that, we should be able to know the signs and symptoms of poisoning. What signs and symptoms you are able to recognize? Okay, these signs can occur in this type of toxicity. Suppose arsenic, okay, there will be gastrointestinal problems will be there. The diarrhea will be rice water diarrhea. Like this, we know or should be able to know the pathognomic signs which is in the system. Apart from that, also we should be able to know the perfect diagnostic test, and we should be able to isolate and purify the particular toxin. Then only we can be present in the court of law. Many a times people when they were first trying to see what is the presence of toxin, how the presence of toxin. So here are some instances when these toxins were being discovered. Okay, these toxins are also causing toxicity and also causing deleterious effect, and through which the person may also die. So these are two instances where white arsenic is being converted to arsenic acid by chlorine water, and there was exposure to this poisonous arsen gas. Okay, so this gas when come, it was being deposited. So they were able to know, okay, there was arsenic toxicity. Similarly, through the stomach content and urine content in 1821, Sarlas were able to understand that there is present of arsen, and they were saying, okay, from this compound also we are able to know what is the toxicity or which toxicity has occurred. Similarly, also they have found that this in the body tissue also, or in the liver, kidney, or blood, there is amount of toxin and the toxicity has occurred. Over the period of time, this MJB Orifella from who have in 1814, he is also the father of toxicology, and he this traced these poisons. He was the first approach to the poisons because then they were not able to define if poison or toxin is causing something very definitely. So after them, they were this later also he is a good scientist in our toxicology. He was able to detect some tests that are if there are tests, they will be simple chemical test, but they will say that okay, this is positively presence of this metal toxin or this non-metal toxin, and this might have caused the toxicity or the death. Many times, many murder trials are there where they were able to understand no arsenic was present in the surrounding of the person, and the poisoner might have given him arsenic for the death of the patient. Next one. Okay. This isolation of the alkaloidal poison. Alkaloidal poisons means it is present in a plant. Plants compound, they have alkaloids in them. This is alkaloid poison. This is nicotine in through which there was a murder and they were able to isolate and able to know that this was the presence of nicotine and the death has occurred out of it. So generally we also have many toxins like colchicine and this quinine. Quinine is from Conium maculatus. We have on that also caused death instantly. And this morphine. Morphine is also treated as a narcotics, or we can also taking we are in the cough syrups also. We also have some amount of morphine. In the analgesics also we have morphine. Also it is a thing of abuse. Also in large quantity or if some more amount of morphine is taken, then it can cause toxicity. Then narcotin, it is also it is a substance of abuse. It is also causing toxicity. Strychnine, it is found in the tree Strychnia noxvomica, and it causes this glycine receptor blocker, and it causes the toxicity in which the person become arched, and it is a very violent toxin. In some years ago, they for killing of these dogs in Canada, they were allowing strychnine, but they were having so violent death that it was banned. Strychnine was banned for to cause any. If we are going for this euthanasia, also strychnine is being bad because it causes a very violent death. We can't give death to any animal with using strychnine. Then slowly, Witheras and Tracy Sebecker, they did some work on medical jurisprudence, forensic medicine, and toxicology, and published the first forensic textbook in United States. In 1918, in New York, there is appointment of Getter, who was did the starting of this forensic toxicology. As I told, he had established many tests, also many books in which the tests are diagnostic for this our toxins. Also in 1949, American Academy of Forensic Sciences was established, and there was practice of this legal forensic medicines. And several other bodies also came up slowly, like the Society of Forensic Toxicologists and the Californian Association were formed, where they were able to form a society. They were able to talk on these toxins and their deleterious effect and how to get out of it, how to diagnose it, how to prove it in the court of law that this toxicity has occurred. Also, in 1975, another board, American Board of Forensic Toxicology also came. It is also to certify forensic toxicologist. In 1998, the ABFT became accreditation program for forensic toxicology laboratories, and AFBT accredited laboratories came up. They wanted to have this qualified and experienced personnel, and they used to give them certification after training them, and the sound procedures for handling of evidence, analysis of suspense, and reporting of results. This should be taught because how nicely we handle the evidence will be preserved for next test. Suppose in one round of test we are not able to get the result. If it is preserved nicely, in second round also you can get the results. So analysis, handling of the evidence is important, and the analysis is also important. When we are analyzing one person, how we should be handling it. There should be no mishandling. There should be no contamination over there to get the perfect results. Also, the reporting of the result. Reporting should be perfect. We should be able to know that what was the condition given over there, given to us, and how after some time the results came and how we have to report the sample size or the sample number. All should be very clear. Next time. Then we will talk about the toxicological investigation in case of a death due to poison. This toxicological investigation, it is divided into three steps. So first of all, we should obtain the case history and the suitable specimen. We should know the surrounding and the specimen which we should collect from the site or the place where there will be, if there is it is a crime scene or if at all it is being presented, we should able to know how to get the specific specimen, what samples we should get. Then the toxicological analysis, where we should analyze what we can get, and then the interpretation of the analytical findings to be confirmed that okay, this type of toxicity has occurred. Next slide. Okay. So first we were talking about the history, how we will take the history. So first, when the patient is being presented to, we should be able to know the age, sex, weight, medical history, if he or she is having some problems preferably, or the occupation, because we are studying or we are listening this occupational toxicology many times, so we should be able to know the occupation of the person or the descendant. Also, we should know able to know if at all there was some treatment was undergoing before the patient was being given to us. The gross autopsy finding or the postmortem lesions or the postmortem findings grossly when we are seeing it, when we are going on opening the body, what were the things like many times when while opening up the body we can also get some smells. With the smells also we can able to know that this type of toxicity might have occurred, and whatever along with the onset of the symptoms has come, and the time of death, onset of symptoms also with the each symptoms changes with time. If the post in the postmortem when the we are getting the body, we can see the passage of time, because over period of time this rigor mortis occurs, also the blood changes occur, so with that also we can know the time of the death. Many times in the postmortem toxicology laboratory, we are getting poisons like over-the-counter drugs. So I should talk a bit about the over-the-counter drugs. Over-the-counter drugs are the drugs or the medication which we can get very easily. We can ask the doctor about this. I'm getting some pain. I need a painkiller or analgesic. They will give you. We can get anti-allergic if you are having cough or a slight cough, or you are sneezing, or you are having a runny nose. We can go for this antihistaminic or H1 receptor blocker. That also we can get from the pharmacist itself. Often you're saying, if the layman is saying and getting the drug, that is the over-the-counter drug. Else, other the drugs are scheduled drugs or prescribed drugs, which only can be given upon the prescription of a doctor. Without a prescription, they can be given. Those drugs are the benzodiazepines or the sedatives or the opiates or the higher analgesic groups. These drugs cannot be given to the person without a proper prescription. And the drugs of abuse like this cocaine, marijuana, and methamphetamine, these are also over present there. And some presence of some gases like inhalant gases will be there, and this carbon monoxide over will be there. These all the things that can be found while we are performing the postmortem of the person. Pathologist or the forensic pathologist, whoever is doing the postmortem, there should be collection of the specimen. The specimen should be collected for various type of analysis. So as per necessary, we will get the body fluids, like the lung fluids we can take. We can take the gastrointestinal fluid also. And also the organ collection, we will go for the liver, we will go for the kidney. Also many cases heart are taken, many cases testes are taken. So these samples will be taken and it will be put in the proper solvent. So many types test we do. So if you are if you want to take the if you want to go for the histopathology, so you will put it in BNF or buffered neutral format. If you want to do some molecular type of thing, then we will take it directly directly we will take it into the 4 degrees Celsius or minus 20 degrees Celsius, like that. It depends what type of test we want to do. For that, sampling is done. We will take that sample as soon as possible to the laboratory and test the test, or we will put in such a solvent that it will be preserved. The particular thing that you want to isolate from that, it will be preserved. Suppose for your molecular, if you want to isolate the DNA level or the RNA isolation, we will put it in triazole. If you just want to see the histopathology, you can put it in the BNF. If you just want to do some test as such, you can just put it directly as such and you can transfer to the laboratory. Whenever we are transferring to the laboratory, we will be having this ice box or some maintenance of some temperature. That further deterioration should not be occurring till we start the test. So detection of the poison is more likely in a tissue in which it accumulates. What it means? It means all the poisons, they have a point of site of predilection or the place where it get accumulates. So many toxins, they will accumulate in the liver itself. If it is gaseous anesthetics or gaseous things, it will get accumulated in your lungs. These vapors or gases, it will be in the lungs. And if it has gone through the ingestion or gone through the kidney itself, the urine sample will be having the poison. So we should be able to know which poison is present in which tissue and how we should collect the tissue, how we should preserve the tissue. So this collection, preservation and dispensing of the particular sample is very, very important so that it will reach the pathologist at the laboratory and he or she will be able to analyze the sample and get the test done and get the poison or the toxin perfectly detected. So when we are collecting the samples also, we should be very much meticulous about labeling of the container. The container, as I told, it will contain the fluid what you want. If you want what the solvent, if which you want to carry, that it will be having already. It should be labeled beforehand. It's not that you will get the sample and then you will try to label. You know what sample you are suspecting for, so you label it properly. You put the date, you put the time of postmortem, and you put the name of the descendant, which person you are taking, the identity of the sample. In some cases, identification number will be there, and if at all possible, signature will be. So it should be perfectly labeled. The labeling is very important when we are going for forensic cases. Many times we see that autolytic or putrefactive changes may occur. So this reduces the specimen quality. What happens with the passage of time, particular these GI contents, that start deteriorating. So many times there will be bloating of the animal our patient. The bloating occurs, and this bacteria they will try to pass out throughout the body. If it is going and passing into throughout the body, so more putrefactive changes will occur. And before the changes would have been occurred if the postmortem is occurring in a room temperature or in some hot humid climate, so more of these autolytic or putrefactive changes are occurring. So we should store it in such a place that these putrefaction changes are very less, and before that we should be able to take out our inference. Either we should be take the samples of the patient, or the samples should be taken out of the patients, or by seeing the condition of the patient, we should be able to detect which toxin might have been occurred. These changes were there. Some you might be seeing some this on natural orifices were having some fluid things. So we can estimate that this type of toxicity might have occurred. So we should be able to store the body or these fluids in a very particular manner till it reaches the forensic toxicologist, and he or she is able to do the diagnosis that what toxicity or what caused the death. Next slide. Okay, so as we are talking that how should be there should be the collection, and the sample should be dispatched to the laboratory so that the perfect diagnosis can occur. So these fluids and tissues, whatever is there, we want to collect is our gastrointestinal contents is there, some urine is there, some in the liver also. In the liver, we are going to take some tissue, and all of them has to be put in some solvent and has to be sent. So whatever is the in the machines we are going to work, each particular machine will need a particular type. So if we are putting the sample in that particular solvent and we are sending, it is very good. If at all in the field level the particular sample is not present, particular solvent is not present, so generally we advise that we can put it in normal saline and send the sample. So generally, our forensic toxicology laboratories, we receive our this blood, urine, liver, tissue, stomach contents, or the gastrointestinal contents for identification. So from these samples, we have to find out the toxins that are present all over there. So this many times also these are the two type of tissues we are having. We are having the vascular tissues. As I have talked, this liver, blood, or urine, all these are vascular tissues. But many times we also go for non-traditional or a different type of things like your bone marrow or hair, nail. Many times we see that in case of some toxins, the more of this amount is being deposited in your hair. Like arsenic, we know the example where the we found the hair after a very long period of time, and still we are able to find the presence of arsenic in their hair, and we're able to know that this person has been poisoned from of a chronic arsenic poisoning, and death has occurred. So for that, we are able to know the amount of toxin present. So also we are using bone marrow, hair and nails. And sometimes in some cases, some putrefaction might have occurred. Many cases in toxic cases, some burning type of things or some deterioration also we see. But still, if we are getting this bone marrow or the bone washing from the skeletal remains also, from that also we are able to detect the amount of genomatics or the toxins present, and that can be used for establishment of this toxicity. Many times this vitreous humor of the eye, it is also secured, and we are also able to get the amount of poison. Mostly it is useful for this volatile poison detection, like the alcohols, ketones or glycos, they are being detected. Here it is mainly, as I have talked, it is for the heavy metals like the arsenic, mercury, lead, they get deposited in there. Also, many times some pharmaceutical drugs or pesticides, plastics also they get deposited, and they can be analyzed, and we can see that this type of toxicity has occurred. This analysis of hair, it is done for many forensic settings after this postmortem. Nails are also used for the for particular some genomatics in both antemortem or postmortem. Also in the nail spaces, also you might have seen that sometimes some remnants will be there, and they can also be taken out, and these scrapings can also be used for this detection of the poisons or toxins. Next slide. This quantitative amount in the toxin present in our hair or nail, it can also say that how much period of time the patient or the dead person has been exposed to those genobiotics, and how much amount is present. With that we can calculate. Finally, in many cases, a decomposed body also comes. From that, there may be blood, there may not be blood, and there's tissues also. They might have got putrefied, and we may not be getting. So many times also, fly larva or the maggots are there. Na? From the maggots also we can calculate since how much time this person has died and this maggots has developed. So maggots also we can take for the estimation of the time, and the maggots also belong to different. Generally, these are the fly larvae, and the flies will be over there and laying their eggs, and from that these larvae are coming out. So by estimating the type of flies present over there, we can also get some information where the death occurred, how it occurred. That also we can able to understand. And the amount, the way the descendant tissue in which the larva were feeding, the amount it got deteriorated, that also gives some idea about the death of the person. Then we can go for the amount of these amount drugs. If at all, if it has died of some sedations or sedatives it has taken, like the barbiturates, benzodiazepines, phenothiazines, that also can be detected very normally in the laboratories. Many times they have been in depressive patients, they have been advised by the doctor itself. But in case of some homicidal or suicidal tendency, they will take these drugs in large amount, and they will it will, when taken in larger amount, these drugs will cause toxicity and in many cases death of the person also. Many times also in these maggots are there, this is fly larva maggots are there. They are when they are present in that particular thing, we are able to know that okay, this accumulation of the particular compound was there, and with this maggot in the maggot itself, if we get the concentration of the drug like propyclin or amitriptyline or nordazepam, it is a metabolite of diazepam. So we are not getting the diazepam directly, but we are getting the metabolite of diazepam, that too in the maggot. Then also we can also know that he has taken diazepam, which is a antidepressant drug, and death might have occurred due to it. Next slide. Okay, we were talking about the process in which we are talking of three steps. First one, the case history we have taken, how the take history or the way the samples have come, like that we were discussing. Now the toxical analysis. Now the samples we have taken, the history we have taken. Now we will talk about the analysis. Before the analysis, we should be know the amount of specimen available. So what amount of specimen that became available for me, and what are the poisons I'm thinking it might be present over there. What are the possible steps of biotransformation which the poison has undergone till the sample came to the laboratory. So many times when there is oral administration of the poisons, the gastrointestinal contents are analyzed because it will be in the gastrointestine itself. The some amount of unabsorbed poison might be present. Some might have gone into the liver or the blood cell. But in case of oral administrations, unabsorbed poisons are much expected from the GI contents. Urine is analyzed next because kidney is the organ of excretion and high concentration of toxicant is there, it might have gone into the urine itself. Then it goes to the liver and get into the general circulation. So we should analyze the liver then. So if we are suspecting for a specific poison or the specific toxin which has contributed to the death, the toxicologist will analyze those fluids and those tissues in which we are thinking it is the way of metabolism or it is the way of excretion. These tissues should be collected and we can test for it. In some instances, the metabolite, as we are talking this in intermediate stage, the toxin will be thing and it will be excreted in another form. But the intermediate state, that also, if you are able to understand it, this is the intermediate state, and we can know that this is the intermediate state which was also causing the deleterious effect. The poison must have caused some deleterious effect along with that the metabolites as well. So we should be understanding what is the metabolite of which drug, which was the main poison and which is the metabolite. Both should be understood by the forensic toxicologist. Many times we also see there are some assays, they will not able to detect the parent drug, but they will be able to detect the urinary metabolites. The major thing in which is that this is your leucocaine methyl ester. So the diagnostic, we can go for the urine sample and we can dip and we can know that this compound was present. Okay, if this compound is positive here, that metal was present there or that genetic was present there, which caused the death of the person. Many times we see that when there is co-administration or co-ingestion of two things at a time, there is some hepatic in the liver, there is some biotransformation which causes another compound formation which is more toxic. So that example is if we are co-ingestion of alcohol along with cocaine. So there is hepatic transesterification of this cocaine to form cocaethylene and it is a psychoactive metabolite. So this compound, if it is present over in the body fluids or the tissues, also in the hair, with the estimation of this compound, we can be able to know that this person has taken co-ingestion of this alcohol and cocaine. So we should be able to understand what is cocaethylene. From that, we can understand there was co-ingestion and there was death of the person. So for this, many times we go for GCMS or gas chromatography and liquid chromatography along with the mass spectrophotometry if it is there. So it is GCMS. There is quantification of these compounds. These pesticides or these genovatives all are quantified and we are able to know what compounds was particularly present in the body fluid and what compound caused what deleterious effect. Next slide. Okay. So also GCMS, LCMS we are using, also high resolution MS we are using. With this MS or mass spectrometer, we are able to know the mass. With what is the mass of the with the GCMS, we are able to know the structure or the particular compound. So we are able to come to the point that this amount of this toxin or this genomatic have in the body of the animal. So this toxicity has occurred. So many versatile screening methods are coming up where these analytes are being used to for detection. So generally, we are taking that the normal chemical changes which is occurring after the decomposition of the cadaver, or we can go for the autopsy when we take the toxicology analysis. Many times we can also see that in the body of the body, when there is death of the patient, so normally there are some enzymatic and non-enzymatic processes, and there are this decomposition process will start, and then the microbial metabolism will occur. This microbial mechanism, see, in the body itself we have several enzymes. So once there is death, these enzymes will also start deteriorating. They are deteriorating and they are also capable of deteriorating these toxins or any poison which was present at the time of the death. So if it was present in the time of the death, due to this enzymatic reactions also, their physical and chemical properties will be deteriorated. So if that compound is going on deteriorating, it becomes difficult for the detection of that particular poison. So, and as earlier 1870, cadaveric alkaloids were being isolated from the organs of putrefied bodies, and they produced some color test reactions which are produced by morphine and other drugs. So, they were able to know that from this bacterial decarboxylation of the amino acid, or like ornithine and lysine, they are able to produce putrescine and cadaverine. Okay, so these putrescine and cadaverine, if they are present, we are able to understand that bacterial decarboxylation occurred of the drugs, morphine and other drugs. So we should be able to know which compounds are we are getting at last. Before that, what compound was majorly present, which caused the death. And after that, putrefaction occurred, and are getting these compounds. So we should be able to understand which compounds were. We should be able to recognize what compounds which can be present if the toxicity has occurred and enzymatic decarboxylation has occurred. Similarly, many cases phenylalanine it is getting converted to phenylethylamine. It also has properties similar to amphetamine, and they will show the different type of symptoms. Hydrolysis, oxidation, reduction of this protein nucleic acid occurs, and there will be changes. And there may be presence of compounds like tryptamine and norhamine. And this causes your psychological type or psychoactive type of compounds. These are psychoactive type of compounds which will show deleterious effect. So these substances also interfere in the isolation, identification, and quantification of the toxicants. The concentration of cyanide or ethyl alcohol, carbon monoxide, it may be decreased because there are some enzymatic reactions which are going on. So the normal concentration of the toxicants like cyanide, ethyl alcohol, or carbon dioxide, it will be decreased, and we will not be able to know what was present at the starting time. So we should also be able to calculate the degree of putrefaction and the microbial activity which has occurred, and this has decreased. So, but in some cases like arsenic, barbiturates, mercury, and strychnine, they are stable, and they are also be able to detect after many years of death. So it depends upon the type of poison or the type of toxicant what is there. So it might be broken down into some other metabolite, or it might be stable as such after many years. So being a veterinary toxicology, being toxicology, we should be able to know which toxin is there or which has decomposed or which has changed. Next one. As we were talking, when we are taking the solvent to take the sample from the body of the cadaver, so the reagents which we are using for test, the solvent in which we are carrying the tissue sample, it should be highest grade possible because if at all it is having any contaminant, so it will interfere with the result. For that reason, we should have the highest grade possible of the solvents or the reagent, and it should be free of any contamination because any contamination may interfere or distort our natural finding, which we need to detect the presence of the particular zoonotic or particular metal, non-metal, or any toxins that was present over there. Like this chloroform, it contains phosgene and ethyl chloroformate. It may react with your primary or secondary drugs to form carbonyl chloride and ethyl carbamate derivatives. So our containers, it should also be free of contaminants. It shouldn't be having anything that will interfere the results with our chromatographic or spectrophotometric tests. So, or they are GCMS, these are all chromatographic tests. So, if at all there is any change in this wavelength of this color change will be there, it will not be detected very nicely. So, we should be always very aware. These are sample containers, lead stopper, all are free of any contaminants. Generally, all the analytical laboratory they will not allow the sample being collected in any container. They will need that it should be collected in this particular GCMS sample container or LCMS sample container. Those are having these sample containers are particularly free from any of this our any derivatives or any contaminants. So also when the sample has come to the laboratory environment, our laboratory should be very clean, and we should every time each laboratory it should be having your its own laboratory practices or do what we say GLP or good laboratory practices. Everything in a lab should be in a particular place. Everything should have a place. Everything should be in its place. This is the place of glassware. This is the place of reagent. This is the place of chemicals. This is the wash basin. This is where we wash the glassware, this is where we wash our hands, this is where we get sanitized. So every place should be properly labeled, every equipment should be properly labeled, or every equipment should be having your SOP or standard operating procedure, where everyone should be able to know in the equipment where he or she is working, what are the steps, how we will work, what is the sample, where I have to mix it, how much amount I have to take, what amount I have to mix, and we can do the test very perfectly. So when we are concerned about the analysis of the metals or some environmental target contaminants, if at all our laboratory is contaminated itself, we will not be able to know the amount of that particular contaminant. So first of all, our laboratory should be clean enough. It itself should not be having any contaminant. This forensic laboratory, they will be analyzing there with their very own procedure. They will be having their specific test. So the specific test will be this for a specific urine test is a ferrichloride set, perchloric acid, and nitric acid. There will be color things for the phenothiazine drugs, and for the detection of amphetamine or benzodiazepine or opioids. There are different tests are there. Positive results are obtained, and then we can go for a secondary procedure and we can identify the particular drug. Nowadays, GCMS and LCMS are most widely accepted because they give you the particular structure of that contaminant, what was present, what caused the toxicity, what was the particular toxin. So we are able to know its mass spectrum, we are able to know its mass, we are able to know its structure. So that is the very particular equipment used nowadays. Analytical methods, it should be very much accurate. It should give your qualitative and quantitative data, which was present, what was not present, what was present, if present, in how much amount which was present. So the lowest control concentration we should be able to dictate in that particular sample. So very precision models are there. They are able to detect very little amount of things also. So whoever is working in the laboratory, they should work with very accurately, with very precision, linearity, and specificity. They should be very particular that I should get, if at all any poison or any toxin is present, I should get the result. So if the unusual sample is coming like the bone marrow, fingernails, head, and maggots are coming, so the extraction efficiency will be more complex because the matrix is different. So different in calibrated, different controls will come. So the laboratory technician, he or she should be also be able to know what should be the matrix, what should be the change, what is the vascular tissue, what is the non-vascular tissue, and what should be the different solvent in different cases, and how we will be able to detect a particular toxin or poison in which case. Next slide, ma'am. This is the third phase where the first one was case history. Then we talked about how we will analyze the thing, how to precautions were. This is the third or interpretation of the analytical results. What results were good and how we will be able to interpret them. This toxicologist should be able to interpret his or her findings about the physiological or behavioral effect of the toxicants on the descendant and the concentration found. So first we should be able to know the route of administration, in which route the toxicant has got into the body. So we should be able to know if at all it has taken orally, if at all he or she has inhaled that thing, or if at all it was a dose. If in case of drugs it happens, it's a dose administered, how much amount he has taken. But after that, how much toxicant was present, which caused the death, or some deleterious effect, or whatever the deleterious effect came to it, how it came. So the highest concentration generally it is found at the site of the administration. So generally, if there is ingestion of these drugs and poisons, so we will get it in the GIT and the liver. If we are getting the high concentration in the GIT and the liver, so we get indicated that it was oral ingestion. So if you are getting high concentration in the lungs or the visceral organs, we can say through inhalation it came, or through intravenous ingestion also this toxin came into the body of the patient. Generally, the relative distribution of the drug, it differentiates the oral from parenteral. From the if it is parenteral route, it will go directly to the bloodstream. If it is oral route, it will go through the liver and it will undergo this your hepatic metabolism, intestinal metabolism, pass effect through where the liver metabolizes before it gets to the bloodstream. Many times it goes to the intestine, again comes into the liver, again goes to the blood. It is intestinal pass effect. So both the processes are there in case of oral administration, and the concentration of that toxin get decreased. But in case of intravenous injection, it gets directly and the concentration remains as such. There is no first pass effect. And smoking, it is another administration for the route of abusers, where illicit things like cocaine, heroin, marijuana, phencyclidine, these are even taken. Paralysis of these drugs also occurs after the inhalation, but the problem is happens when the cocaine smoking, it got that crack drug or anhydrocannabinol ester. Thus, if we are able to identify the high concentration in this compound, so we are able to know there was cocaine inhalation. We should be, as I've talked, we should be able to know the metabolite which has come. We should be able to recognize, okay, there was the indication that the smoking was there and the route of cocaine administration was through smoking. And ultimately, we are getting this cocaine metabolite in the urine. Next slide, ma'am. In the GIT or the gastrointestinal, if there is presence of a toxic material, regardless of the quantity, we should be able to know which agent caused the death. We should be able to demonstrate the absorption of the toxicant. This the absorption has occurred and it has gone to the general blood circulation and the target organ where it has exerted its lethal effect. So by the blood and the tissue analysis, we are able to know this. Also, if if some strong and corrosive chemicals has come, like the sulfuric acid, like phenol, it is directly destroying the tissue. It need not go to the system, go to the liver, or go to the lung, or go to the kidney and show its toxicity. It is directly destroying the tissues because it is corrosive itself. The point of ingestion also, it is digesting the tissue cut, causing the hemorrhages or shock. Generally, if we are going for the urine analysis, so the physiological effect of the toxic agent will be known, because if it has gone to the body till the urine, the animal has ingested it, and the urine established that the poison was present sometime before the death, because the urine toxin was present, toxin got into the body, and then it went to the kidney, it is deposited in the bladder and formation of urine. Urine is having the toxin, that means it was present in the body of the animal before, sometime before the death of the animal. So correlation of the urea value with the physiological effect can be done or cannot be done, because excretion of different compounds, it occurs with different time and different urine volumes also. Generally, most drugs and poisons are correlated with the blood and the blood fractions. With the we can take the plasma for testing, we can take the serum for testing, and we are getting the better therapeutic drug monitoring. The postmortem blood is resembling from the vasculature after the death. Thus, the interpretation of the postmortem results careful consideration of the case history, site of collection, and postmortem changes. We should be able to know the death after how much time we are getting the postmortem sample and what symptoms we are getting, or how much degree of putrefaction have occurred, and the site from which the sample has been collected, and the case history where the death has occurred, what was the circumstance, and what circumstances the evidences also have been collected. Like, next slide. Many times, blood values, it may appear that it is non-toxic, that there is not much amount of toxin present in the blood. But it can happen that the death has occurred in some such cases due to hepatic failure, due to acetaminophen overdoses. So that concentration may be consistent for therapeutic doses, but the death might have got due to hepatic failure. The many times we should go for this case history, and we can go for histopathology. In the histopathology, we can see that the necrosis of the liver has occurred and the central lobar necrosis has occurred. And also we can get the serum specimens from the diseased person, and when he or she was in the emergency department, and we can get the samples also. Also, due to the medical emergency treatments, like they will administer the fluid, plasma extenders, bicarbonates, diuretics, blood transfusion, it dilutes and removes the toxic agent, because they are treating the person. The person should be treated so slowly, there should be elimination of the toxins from the body of the patient. So generally, upon prolonged survival, or after he or she has gone this all the respiration or this hemodialysis, hemoperfusion, so it decreases the amount of lethal blood concentration in the blood. The poison amount is getting decreased because they are diluting it. They are trying to move out the poison from the body of the patient. It is generally assumed that the postmortem blood drug concentration are more or less uniform throughout the body. However, in 1970s, they have found that the concentration of digoxin in heart blood is excreted to the from collected from the femoral blood, because we know digoxin is a drug acting on the heart. We are getting it for myocardial infection. So they have seen that in the after the postmortem, digoxin is much present in the heart blood than in the femoral blood. Also, in some cases, they saw that, particularly in the heart blood, their values were particularly changed. Also, they have seen that in the subclavian vein or the thoracic aorta, inferior vena cava, femoral vein, so forth, all the different blood vessels are having different concentration of this particular poison with which the death has occurred. So, different postmortem blood from different side, it will be having different amount of particular drug which you are calculating, because toxins, they are of different nature, and its concentration in different blood samples are different. So the site of collection of blood is very also becomes very important at this point of time, where we are seeing the particular compound is more present or less present in a particular type of blood vessel. So it becomes site dependent also. Also, the interval is very much important, because with the interval of the specimen collection and the death, particularly in the heart blood, much changes are occurring. When the heart is stopping beating or heart is having some problem, so there is much change in the heart blood. And over the period of this postmortem interval, the basic drugs with having more volume of distribution, like tricyclic antidepressant, they will just be more distributed, and less amount will be present for their detection. Next slide. So this analysis of tissue specimen is very important, because we should know the minimal administered dose, or at all it became a burden for this drug or poison. The concentration of drug in each specimen, there are some calculations which we can know in which particular tissue we have found, with which particular fluid we have found. In that, how much we have found, and we will calculate with the total body weight, and we will get the how much amount of toxin was actually or really administered. So we can find the difference in the fluid and tissue, and totally we can calculate the whole body how much burden was on it. And this approach will be very good in solving the medical legal cases. Many times in case of arrhythmias, there are poor circulation and poor tissue perfusion. So this lidocaine, it is also an antiarrhythmic drug. It will not be well distributed in case of a fatal heart attack. So postmortem blood, it will be having 50 mg per ml in such cases. For value for antiarrhythmic does not exceed 5 mg per ml. This difference occurs because of this, because the patient itself was suffering from arrhythmia. Itself, the heartbeat was not proper. So when this perfusion of this blood concentration, it differs. So generally there will be not particular assessment of the particular amount in a particular case of point of time. So it becomes artifact, and so it may go for a fatal overdose. But this we shouldn't take that the dose was not okay. The dose might be okay, the distribution is not okay, because the patient was suffering from cardiac arrhythmia. Also, if you see our non-vascular tissues like hair and nail, so it has a longer window of detection, because it is not having a very good blood circulation. So these toxins are not passing away very fast. So whatever is being deposited in our hair or nail, so there is permanent retention or long-term retention of these drugs, even when the plasma and the urine concentration we are not able to detect because it has passed away. But in this hair or nail, it will be deposited as such, and we can detect it after a very long period of time also. Many times we can see in the content of the root, there is significant amount of arsenic. And this occurs because of the presence of these germinal cells. Germinal cells are present on the root of the hair, and so the circulating arsenic gets more deposited, and the concentration in the blood may rise or fall, but in the growing hair the concentration will be as such and more than the blood or serum. Next slide, ma'am. So we now see why our forensic toxicology becomes important. So this poisoning, if it is a criminal poisoning of the living person, so we should be able to know what are the toxins being given in case of a live person who are being uh, treated with those drugs or these xenobiotics or these poisons for causing some crime. So generally, if they are giving they intoxicate people or kids with this alcohol and victimize them with your kidnapping or robbery or sexual assault can be there. So generally, we can see that in the when there are people they are getting slaves or something like that, they will be making them drunk and they are not able to know that they are drunk and they will be welling over period of time. Particular to this concentration, what happens because they have taken much of the alcohol or much of the anesthetic is there, so they are able to procure these persons from one country to another and then they are able to sell those persons also. So when administered this alcohol are administered without the person or knowing, or sometimes the person was going to getting intoxicated to certain level, but they have fed them or drank him with more amount. So he or she was not able to remember what happened to them. Many times amnesia occurred, and when they are drugged, they undergo severe depressions also, and it becomes very difficult for the toxicologist to know what toxin or what they have been given, so that they have forgotten what happened to them. Next slide. There is another syndrome called Monson syndrome. It is a condition where the patients they seek admissions to and they say that we have medical histories and we need some medical attention. Generally, these patients they need some attention to themselves. So what happens if they are having a kid or their child? They will tell that our child is ill, so for this purpose, they will be giving some compounds or something to the child itself, and the child will be showing some symptoms. Actually, this not be might not be the child may not be ill, but because of feeding of some, they are giving some deleterious compound. So this he or she or the kid they will bring that he has to be hospitalized just for the sake of attention. They are feeding that, but the problem occurs when they are taking these things. Generally, in such cases, they are giving imetine or cephalin, but occurring in the syrup of epicar. But in some cases, it becomes more, and even death of the kid is also seen. So, in such cases, if some forensic experience is there, we are able to know that this syndrome has occurred, and we can go for legal proceedings also. Next slide. So, we will next go to forensic drug testing. So these forensic testings we all need, because in case of some poisonings also we are needing these services. But mainly in case of this military services, it is a very aware area. So they always need this transportation of this their arms, ammunition. Also, in case of nuclear industries, also they are having their participants, or the persons will be working in inside their laboratories, or the persons were engaged in the storage of the things, or all the people over there. And also in case of many federal state agencies also, where they are working with our poison substances or any substances that can be harmful, there these testings are being routinely done, because a toxicity or poisoning is a very likely occurrence there, because they are working with this see, in case of our communications they are working with this gunpowder. So the people who are handling, who are taking, who are storing, whoever they are making, so all the people they are having natural exposure to it for long period of time. So in those cases, so these tests are being done very routinely, so that these people who are working on the system, they shouldn't be getting poisoned or shouldn't be getting toxicity out of the things they are working with for the better of this mankind or for better of us all. Also, many times we get that well, there are problems where one party is saying that this toxicity has occurred for this persons, but many times the employer they will say no, I have taken all the precautions, there shouldn't be this type of toxicity. So many times these types of legal complications occurs, and we need to see what has actually occurred, if the person is suffering from toxicity or poisoning for this particular condition itself, or it might be the both the sides, they can be the problematic. Either the employer, he or she has not maintained that the toxicity or poisoning shouldn't be there, that is one case, or else this person is working here but may be getting poisoned due to some other toxin. So many a times there should be very legal cases where either if the person is saying untruth, so he can be court-martialed or dismissed from the job. Either the employer or the employee, both can be at fault. So we should be able to know what actually the poisoning occurred, or why he or she is suffering, what are the symptoms, what are the amount of toxins present over there, why he or she is suffering. So this has to be very much legalized, and we should be very perfect in diagnosing all those things, because this case become legal cases. For this testing, so there are many certification programs. So laboratories are conducting testing of federal employees are required to certified under the Department of Health and Human Services, and there are mandatory guidelines for the workplace of drug testing as published in April 11, 1988 Federal Register, which has been periodically revised, and the latest revision was done in January 23, 2017 Federal Register, where HS is allowed collection of an alternate specimen such as oral fluids by the Department of Health and Human Services 1988-2007. The College of American Pathologists also conducts a certification program for urine testing drug laboratories. So our government itself is very aware, and they understand that this forensic pathology is very important for detection of any kind of crime or any kind of occupational hazard. There is a certification courses so that everybody is able to know the perfect thing and we can act perfect. Next time. So many times who are in those employed in those conditions only, they are usually given or regulated for urine testing very regularly. So if a person wants to change the result, if we are see he or she, they are going undergoing every time they are undergoing test, they know that in the test they will be caught if at all they have done something mischievous, and they will be caught. So many a times they have used drugs which by which this test will not show positive results. So these techniques or these drugs are used to mask their drugs which they have taken for their use, or for their for intoxicating themselves or getting away from any kind of things. So what they use diuretics. Diuretics means the drugs which will speed up the urination process. So if you have taken some type of this intoxicating metals and you are taking diuretics, and the urine will be passing more fluid formation, more urine formation will be there, and your urine will be taking all these metals very quickly away from your body, and you will not be caught. Also, they do what they do, they adulterate the specimen directly, because we are nowadays we see the many crime serials or episodes where we see that think that they have taken something other thing. The sample is saying, but many movies you might have seen that yeah, the sample was stored that, and the people have changed the sample itself. The person who knows that I'm going to be caught by this process. Many times they are doing DNA fingerprinting, they are doing many tests they are doing, and the person who knows that I'm at fault, I will be caught. They what they do, they will try to mask. They will adulterate the specimen. They will change the specimen. Many times it comes in news that the specimen itself was changed. That specimen, if at all it is being changed, we can't get the good result, we can't find the culprit. So what they are doing, they're adulterating the specimen itself. If they put bleach, if they put vinegar, so it will really hamper the immunoassay test, and we will not get the result what we want, or some criminal or culprit, they will escape through this adulteration or changing of this specimen directly. So these specimens are should be routinely tested. If you want that these specimens should be preserved for long period of time, after 1 year or something, we again want to test it. So we should be checked for the adulteration. So we should check that it is not being changed. Its specific gravity or its presence of this whatever normally was there in that urine sample, it is there, and there is no change in this usual color or smell. If at all is there some change, that means it has been adulterated or it has been changed. So for these, many industries are also developed to beat the drug test, because Many times in case of doping, you might have heard these athletes, they take some drugs to have some energy, stamina at the particular time when they are going to perform. So they will take this drug, and also they want that the drug shouldn't be caught in their blood, else they will be expelled from this sports life throughout their life. So they, to beat the drug itself, they are getting chemicals, and when it is being added to the urine specimen, it is not allowing the confirmative drug test, and we are not able to know the particular amount of drug, if at all it is present there, if at all it is not present there. So many times, other than substance of abuse, also we can get also positive findings due to some therapeutic drug control use of these controlled substances. Some drugs might be going inside through the food or this was passive inhalation. For example, if you see the seed of Papaver somniferum, poppy seed, it is a common ingredient in many pastries, breads, or our household dishes also contains this some amount of this poppy seeds. But this urinary excretion may have some amount of morphine, but that doesn't change make the fact that there is morphine ingestion. Many times also that positively due to our normal food habits also we can get some metabolites metabolites of heroin is also morphine, but by taking our normal food also we can get morphine in our urine. So many a times it is very difficult to really differentiate if it is heroin abuse or it is a normal poppy seed ingestion. So analysis we should do for that particular drug which we are suspecting. If we are suspecting for the heroin, so we should search for that particular heroin metabolite so that we can rule out it is not normal poppy seed ingestion, but it is particularly heroin, and this is the heroin metabolite. And we should see for 6-HTL morphine, not only morphine. Then only we can rule out that it is not some normal culinary ingredient which has been taken in the food, but it is usually a heroin metabolite, and there was personal drug abuse. Next slide. So there can be two terminologies like forensic drug testing and forensic toxicology. So this forensic drug testing, it is a normal test which is generally done many times. But if we go for forensic toxicology, it becomes very particular, and there is a particular matrix for which we do the forensic drug testings, and we should be doing this very meticulously. And this forensic toxicology, it is generally always on emergency basis. So more alternative metrics, more funds will be there where the collection will be more convenient. In less amount of time, we need the results. So this forensic toxicology, it will be in an emergency situation, and forensic drug testing, it is generally done as such. Slow results can also work, but in forensic toxicology, we need immediate results, immediate answers. So more funds we are putting in this to get a convenient and less invasive result. To handle this work workload, many tests we are doing, which is rapid and high thorough output is coming. So this HSS certified laboratories, they are performing our GCMS or LCMSMS. So these are giving a result very nicely and very confidently. The compounds are coming out, and we are able to know this poisoning or this pesticide toxicity or this metal toxicity. It has happened in the patient itself. This laboratory operation, it should be thoroughly documented, starting from the specimen collection and from which person it has been collected, the chain of custody, from which person to which person it came, which lab to which lab it has gone, and the quality control process, how they were collected, which person they were collected. All this should be very particular. Also, these persons who are performing those tests, they must be very well trained, and these methods on which they are working, they should be valid methods, and they should be very experienced in testing those things. This testing procedures should be very finalized ones. These personnel should be qualified, and also the reporting process of reporting of this result, it should be very standardized, and this personnel should know how to report, whom to report the particular results. The laboratory facilities should be constructed and operated in a very fine way so that the total security of the specimens and documents is there. Because we are talking of an era where we are getting on surplus because we are changing the specimen, we are adulterating the specimen. So there should be security of the specimens also, and also the documents. Many times we have seen again in our TV series or in our movies that they are changing the reports, they are bribing the person or something. So there should be very much security till that document reaches the legal authority. How should be the legal authority should know the what is the particular result. So confidentiality is a major thing. So those people who are working in this field, they should know that we should be very loyal. We should be giving the result to this particular person only. We shouldn't be getting molded with some outsider and getting a negative result positive or positive result negative. So this person should know whom to give the result and from whom to receive the result. The authority should know from whom to receive the result, and the person should know whom to give the result. Next slide. So in case of many times we have seen in case of just driving people are there, no? We are seeing that in the roadside also people are there. They are taking the inhalation test. They can see the breath analyzer can see how much amount of alcohol etc. are present. So many a times over period of time people have examined, people have seen how the amount of ethanol or other drugs or chemicals in your blood, breath, and other specimen, and how can they modify your performance and behavior. Many times people that okay I can drive after drinking. It's not the case. Many times there is change in your performance, in your performance is a bit not okay, so that much amount is not allowed while driving. So for that we are doing this test time to time while people are driving. So many applications we have seen under the influence of ethanol or other drugs. Under the influence of drugs, the performance of the people is not okay, and they shouldn't be allowed to drive or they shouldn't be allowed to move in a heavy vehicle or like that. So many times we also calculate the BAC or blood alcohol concentration for diminished driving performance. When we are seeing that the driving performance is very less, so we go for this blood alcohol concentration and we can see that this is the body weight of the person, and after the ingestion of 2 beers within our time, this much should be this concentration in his blood. And through that we can say if he is allowed to drive or he is not allowed. So this ability of analytical methodology to routinely quantify minute concentration of drug in the blood must be established. Everything like this amount of drug, it is present in the blood, this will hamper this performance like this much. So there should be a standardized process, and we should allow this much of blood or we shouldn't allow. We shouldn't allow alcohol consumption and driving like that. There should be tests should be controlled, and the result test should be standardized, and we should allow or we shouldn't allow this person to drive on the highway or expressway like that, which should be very clear. Ma'am, next slide.
Yes, I have changed, ma'am. Even next year.
Okay, okay. Okay, okay. So actually, I talked a bit about this, so I got confused. Okay, if we are able to correlate how much concentration of ethanol is in the blood and what should be the performance of the person, we are able to know the graph or the response what the person should be with. So after the influence of so much of alcohol or such much of drug, if he should be allowed to drive the motor vehicle or he shouldn't be allowed to drive the motor vehicle, that becomes a problem. So it should be very standardized. If at all, if your blood is showing this amount of alcohol or this amount of drug, you shouldn't be allowed to drive. So the people they should be also made aware that even if you are not agreeing, there are many deleterious effects of these drugs, and it will affect your driving performance. You will also face an accident, and the other persons who are also on the road, they will also have a problem. So they should be able to understand. And many times we have seen that this occurrence of the drug in the body of the people, they are resulted in injured drivers, and also some innocent people are also getting killed through this process. So we should see that if the highest incidence drug use or accident rates are increasing, so controlled use of the drugs should be advised. And what these drugs, mainly cocaine, benzodiazepines, the sedative version of phencyclidine, these people are abusing for some kind of intoxication. But under is this influence of drug, driving shouldn't be allowed, and it should be tests, and there should be testing at the point itself, and the legal and scientific problems should be they should be made aware. And concerning the drug concentration at the particular period of time, at the particular point of time, he or she is driving, or he or she is doing some work, and if at all they are intoxicated or under some drug influence, they are not able to work. And that should be standardized that this amount of alcohol or drug is in your blood, you shouldn't be allowed to drive. And that should be very clear, and legal implications should be there. And so, if people are not obeying at all, there should be fines and impairment should be there. If at all they are repeating the same thing time after time. The court testimony, and many a times forensic toxicology they will be called as such. There are two types of witness. There is lay witness. Lay witness means he has just seen whatever occurred at a place, just it is known to her, and he has seen, so he is lay witness. In contrast, if the toxicologist is being called out, he is expert witness. He knows the thing, why it occurred, how it occurred, how much concentration is there, so that it has occurred under the influence of which drug or which toxin. So he is an expert witness. He knows the toxicity, how it will be occurring, what problems it will be occurring. So he will be the successful presentation of a attorney case. So during direct examination, an expert witness, he has to explain the fact, explain the it with a scientific basis. Not that he has known the case and he will explain it as such. Not that the thing. He will be saying it with some evidences, with his normal experience and evidence, how much of this drug concentration is making problem. That should be clear. So with his scientific objectivity, he has to tell the court that this person has done the wrong thing, and this much of drug or intoxicating material or alcohol, whatever, shouldn't be in his blood or her blood when he was there. Next. So in clinical toxicology, analytical toxicology is having a very important role, because if we are able to analyze only, if we are able to know that what amount of a drug is present, then only we can strongly say, okay, this amount of drug or this amount of toxin was present in the blood. So there might be some criminal angle to this case. So, as an aid to diagnosis and treatment of these toxic incidents, as well as if we are monitoring the case, if we are putting the treatment regimens, we should be able to clear that this effective treatment is going on, and this nature of this after this toxic exposure, now the treatment is going on, and the amount of toxin it is getting decreased. So, clinical toxicology laboratory, it will be treating and monitoring the amount of chemical that is remaining in the circulation, and how much is being excreted, how much is still there, what antidote we should give, what fluids should give, should there be given any emetics, should any laxatives be given. So that perfect estimation of the doses, how the treatment should be given from to time to time, because the amount of toxin is being decreased over time to time. So clinically, the clinical toxicologist should see how much toxin is being decreased and what more treatment regimen should be changed. The treatment regimen can be slowly changed with the help of a clinical toxicologist. Apart from the instrumentation and methodology used in clinical toxicology laboratories, the forensic toxicology should be more responsible. He or she should know the case very nicely, and many times he should be giving a legal witness, so he should be very clear about the facts, what he is talking about. These results should be communicated to the clinician who is working, who is treating the patients, and the best methods for this knowing the particular test, for conducting it for maximum accuracy, it should be done by a forensic toxicologist very meticulously. And the main thing is that if, because it is life and death for some people, and some cases crime has also occurred, so it is not a luxury, or it shouldn't be with time we cannot change it. We should be doing it very strictly, because human lives are involved in it. Additionally, because toxical emergencies will come, so clinical laboratories should be giving testing every 24 hours. Now and then, whenever we need, we should be able to assess it. because any poisoning, any crime, it doesn't knock and come, it will come as such. So we should be having these facilities 24/7. Next slide, ma'am. This many a times we have seen that to know this alcohol, to saturate this enzyme alcohol dehydrogenase, it blocks the conversion of methyl alcohol ethylene glycol to the toxic metabolites. So continuous monitoring of these metabolites, we can be able to know if at all it is getting away, getting rid of the toxic metabolites, and we are getting cleared of this intoxicant through the dialysis, and proper infusion is there, and proper antidote is coming. Because for methanol, if it is toxin is be given, ethanol can be replacing it, and it can recover over the period of time. Urine, it is analyzed from time to time for administration of methadone and other therapeutic agents to who do not use to abuse the drug. Many times, psychiatrists and neurologists and the people who are treating the patient for chronic pain, they should also know, because many times when they are getting pain, mostly in case of cancer patients, they get a very high level of pain. Many times they get self-abused with drugs, so they will not get the pain. So they should also know the psychiatric and neurological condition of the people they are treating. So analysis of the drug also we see in the meconium and urine of the neonates to see the withdrawal symptoms in the newborns or fetal exposure to the controlled substances also. So toxic metal determination like blood metal concentrations, it should be also known, and the concentration we will allow us to know the level of severity. With if more concentration, more severe is the condition. If it is slow concentration over a period of time, that chronic toxicity is there. So the period of exposure and the concentration will allow us to know the condition, how the person has got this toxicity. Next one. The factors influencing this therapeutic monitoring, it is depends on the extent of drug absorption and distribution and binding in the body tissues. As for every other drug which we are taking, so there is a distribution of these drugs. The drug get metabolized in our body. Through monitoring this plasma and serum concentration at regular interval, we can know how much amount is left in the body and how much is getting out of the body. So after particular intervals, we should be able to know the plasma drug concentration, and in course of the therapy, whatever we are trying to eliminate these toxins out of the body, how the therapy is getting setting in, and how the effective concentration of the particular antidote is taking place, that we can monitor through this therapeutic monitoring, through this effective concentration of the drug, and through this, if we are going for this kinetics work, and we can able to know how much if the person is recovering. Next one. In cardiac arrhythmia, this procainamide drug is being given, and it is being converted to N-acetyl procainamide. This metabolite has antiarrhythmic activity with equal potency. So many times we see people they metabolize the drugs in different ways. So these fast acetylators are there; they will metabolize in a more faster manner, and the slow acetylators they will metabolize in a less animal. So obsolete characterization is not necessary, because both the drugs, which was in previous thing, which was in latter form, like procainamide was there and NSF, both are having equal potency. So in those cases, particular characterization is not required. But however, if the concentration of the next metabolite is more toxic, so in those cases, we should be able to know which was there and how much it has broken, and how much toxicity can the next compound cause. So all the things should be very commercially known. For these cases, we should be able to assess those commercially available kits for immunoassay, and direct or faster diagnosis of these toxins or poisons. Oh, lastly, we come to the summary. So these analytical techniques, they are being used by our forensic toxicology in expanding the complexity, or having this reliability or sensitivity, because we are able to know the particular concentration of the particular toxin or poison being present, or the genobiotics being present. So with confidence, we can say that, okay, this amount of poisoning has occurred because I got this amount of this toxin in this body of this person. So this is a new field, but upcoming field, because crime are on the rise, and we should be able to understand what amount of toxin can be given deliberately or accidentally, and what the person can suffer. So the drugs of abuses also, it should be known, and the forensic toxicologists, they should be able to identify the toxin in a manner in relation to the analytical toxicology, also in relation to the presence of the signs and symptoms also, and also we can go for this histopathology, and also we can see how the patient is recovering. If the antidote is perfect, the person may recover. So with taking all these things into consideration, along with the health workers or the environmental effects or the history where the person was residing, why come this can, all the things should be summarized in one nutshell, and the toxicopathologist should be able to know what toxin or what poison made the patient come to this stage. Many times the this toxic the analytic thing, if the analysis is very good or very perfect, then your diagnosis is very perfect, and you can go for the therapy immediately, and you can come out of the toxicity.